Hi guys, this is Alicia with Good Morning Sunshine. So I've had 3 lectures so far. I still have 2 more to go, immunology lectures. If I came home, I'm going to listen to those online. But I thought I'd start my video because I have 5 lectures to do today. So we're going to start with hematology and then get into production systems for pigs, so swine. And then we ended with microanatomy of the immune system. So I'm going to start with hematology. So we just got an intro of it. So we're talking about erythrocyte physiology. So we have so that would be red blood cells. So we have the picture of a red blood cells. They're biconcave discs. So we got into the different types. So dog versus cat versus horse versus an alpaca. I don't know why an alpaca is in there, but it is. So we have these 4. Dog being here. Dog has the white in the middle. Cats are a bit smaller than dogs are, so they're over here. And then we have the horse, which are kind of arranged in rows rather than random like the dogs and cats. And they are also smaller than the cats. And then we have the alpaca, which are oval shaped. So I think we're gonna have to know how to identify like which animal will these come from. So erythrocytes are primary for oxygen. Hemoglobin, there's 4 components. So you have 4 perforin rings paired with a globulin protein or chain. You have the iron ring in the center, and it can carry 4 oxygen molecules. So we have to know the important aspects of hemoglobin, and those were the 4 components. So erythropoiesis, we got into nutrients. We have erythropoietin, which is also known as EPO, which have to do with the kidneys and decreased oxygen levels. And then we have iron, which is the formation of the hemoglobin or nuclear division, because it has 2 roles. And then we have iron transport proteins. We have thyroid hormones, T3 and T4. We have vitamin B6, which is pyridoxine. And then we have copper, vitamin B9, which is folate and folic acid. And then we have vitamin B12, also known as cobalamin. And then we have IL-3 and GM-CSF and G-CSF. And they are present in the bone marrow. So that's where they live. That's their environment. So they start out as a rubroblast. So that means they want to become a red blood cell, but that's what they start as. So they're bigger here. I can show you the chart here. So up top is our rubroblast, and then that divides into porobriocytes. Big word. So they get smaller as they go down with each roll they go through. So then that breaks up into and becomes rubrocytes. And then those break up again. So they are rubrocytes from here to here, actually, before they change into the metarubrocyte. And then they turn into a reticulocyte. And then finally the erythrocyte. So an actual red blood cell. So hemoglobin actually increases as the table goes on. That table goes on. And like I said, the cells get smaller. And then they also go from the color of being blue to the color of being red due to hemoglobin increase. And the metarubrocyte is also known as a nucleated red blood cell. So that's a few steps before it actually becomes an actual red blood cell. And yeah. So these are like young to mature cells. So young is in the corner over here, and it goes through steps and then becomes that. So then we have hemolysis, which is normal levels of bilirubin that go out into the feces. So yes. So then we have CBC parameters, also known as red blood cell mass. We have to know this for the exam, I think. So we have to know what packed cell volume, so that's PCV, which we use at work or used at work. And this measures microhematocrit tubes. And then we have the hematocrit itself. So packed cell volume and hematocrit are basically the same thing, or you want them around the same values. And then we have hemoglobin concentration, which is basically 1/3 of the packed cell volume. It is measured by photometric method. And then we have the red blood cell concentration, so the RBC. And that's measured by impedance count. And then we have the mean cell volume, also known as MCV. So basically how big the cell is. And that's measured by impedance. And then we have the mean cell hemoglobin concentration, also known as MCHC. And that's a calculation as well. So feline reticulocytes, there's 2 of them. They have an aggregated one, and then they have a punctuate one. So then he showed us like 2 samples. So a CBC from a small animal, both from small animals. One was hemolyzed, and then this one was dehydrated. But yeah, so the hematocrit should be the same as the packed cell volume, or around the same, roughly. So then we got into leukocyte physiology. It has granulocytes, and then we got into myelopieces or nucleopieces. They have various pools. So they have bone marrow proliferation. They have a storage part. They have a circulating and tissue pool. And this comes from the lymphocytes and macrophages. So again, their nutrients being inflammatory mediators, IL-1 and IL-16, and then lymphocytes and the macrophages, and then the IL-3 G-CSF and GM-CSF. And they also live in the bone marrow, like the red blood cells. But like I said, we just have to know that they have pools. So again, they have a chart as well. So they start as myeloblasts. They divide into pro-myelocyte, become a myelocyte for a while, then become a metamyelocyte, and then they have a band and a segmenter. So these are, again, bigger to smaller. So it takes about 7 days in order to create a leukocyte. And there's about 5,000 to 10,000 normal white blood cells. So leukocytes are also known as white blood cells, if you didn't know that too. A myelocyte, though, is a secondary granule. So again, here we have the picture here, going from young here to older, or more mature, not older, I guess, would be the more correct terms. And then we got into platelet physiology. So morphology, we have between a dog, a cat, horse, and alpaca still. Basically the same here that I showed you earlier. And then we have thromboesis, their nutrients being thrombopoietin, which comes from the liver. And they also live in the bone marrow. So all 3 of these live in the bone marrow. But here they get smaller to bigger, so that's what makes them different from the rest. So platelets, so we have neutrophils that live the longest. Yes, so neutrophils live the longest, and then platelets, and then we have red blood cells. So granulocytes die first. So we have to know that for the exam. And then 5 days is for the life of platelets, and then 100 days is for the red blood cells. And that's all we learned into hematology. So then we got into projection systems of swine being pig. So the swine industry, we have the U.S. hog inventory is slightly up. So as of March 1st of 2021, the number of hogs and pig was 74.8 million. This is down 2% though from March of 2020 and down 3% from December of 2020. Breeding inventory, we have about 6.21 million. Average pigs saved per litter is about 10.94, but it was 11.04 in 2020. And then we have market hog inventory, so we have 68.6 million. So 40% of hogs are under contract-owned operations. That's less than 5,000 a head and raised by contractees. So the swine industry produces pork, the third most often consumed meat in the U.S. So of course we have beef up there, you know. This industry is heavily dependent on feed grains, especially corn, and protein supplements like soybean meal. So where are these pigs located? Well, most of them. We have Illinois, North Carolina, Minnesota, Montana. So as you see, it's the states all that are in blue right there. Probably missed some, but here we are. So changes in the industry over time. Traditionally a corn belt enterprise with farmers, that is crop producers, as the swine producers often utilizing outside facilities or quite extensively. Rapidly moved to vertically integrated companies that own, so vertical integration, the companies own the hogs, they have the feed mill, they have the slaughterhouse, and they have the distribution center. Then we got into facility changes, geographical movement to areas where economic and politically favorable climates exist. Integrator housing is mostly enclosed and climate controlled, so they have a controlled environment. Confinement housing causes more problems with disease, such as parasitology. So we still have like industries like these at the bottom here, this bottom picture. So those are our little industries. Those are our little niche markets, and they're usually used for organic pork. So we have different types of operations for swine. We have farrow to finish, so that's giving birth. Then we have feeder pig producer, which pigs become a certain size and then they are sold out to a contractor. And then we have feeder pig finisher. That's when they go to market. So finish usually means they're going to market, and producer means they usually get them to a certain size and they produce them. So then we have contract producer, we have purebred producers, those are for the genetic companies, and then we have breeding companies, and we have specialty niche markets, which I talked about, and then we have the vertical integration, which I talked about. However, goals are all the same, to produce a healthy pork product for the consumer. So how do we do this? With good management and knowledge, with good relationship with vets, and for healthy animals. So we have the players, which are breeds of swine. Pork producers utilize purebred seed stock of 9 major breeds. So I'm going to show you major breeds right now. So we have the Yorkshire, which is also known as the large white. It has less coarse hair, it has erect ears, and it produces piglets. It's good for milk. The landrace in the bottom here also produces piglet and it's good for milk. Then we have the Duroc, which contribute to muscles, so terminal lines, and they are used for pork, and they're usually males. Hampshire also is for pork and for market, so that's a terminal line as well. And then we have the Berkshire here, we have the Spot, we have the Chester White, and the Poland China. Synthetic lines are derived from these breeds by genetic companies. So the first 4 I talked about, which was the landrace, the Yorkshire, the Duroc, and the Hampshire, we have to know those for the exam. So why have all these breeds? Well, they take advantage of each breed's trait to make the best pig for pork production purposes. Production of a carcass with desirable muscle quality with appropriate palatability. And there are 4 basic cuts into which a pork is separated. So we have the shoulder, the loin, the side, and the leg. So ideal pig characteristics, they're born from a maternal line capable of weaning 25 pigs per year. They reach market weight at the appropriate time and weight using a crossbreeding system. And then meet targets for grow parameters. Though parameters, there's 3 of them. So 21-day adjusted litter weights. Average daily grain is 1.3 to 2.3 pounds wean to finish. And then days to market, the target is 150 days. So 160 plus has interference. So crossbreeding systems, production systems use crossbreeding programs to take advantage of heterosis or hybrid vigor. This is when purebreds are crossed, usually Yorkshire and land races are crossed. Females are selected based on reproductive parameters and bred to terminal sight lines. So those were the early, like Duroc was one of them. And then can select breeding females that are also crossed with two maternal lines for hybrid vigor of those traits. So you have a F1 replacement gilt. So breeding genetics pyramid. So the F1 replacement gilts are young females selected based on reproductive traits of their dams and sires and considered genetically superior. So then you have the great grandparent and nucleus herds. They're raised in multiplier herds and then populate farms where they need females replaced. So those are known as the replacement gilts. These animals can also be used for production of commercial market pigs. So the basic production systems, so we have farrow to finish, which are usually the small operations, farms that involve all stages of production from breeding through finishing to market, weights of about 275 pounds. And then we have farrowing nursery farms, which self or just transfer 40 to 60 pound feeder pigs to grow finish farms. So this is where they're in the nursery period until they go off to the weaning and the bigger farms so they can get to market. And then we have farrow to wean, so get bigger in the farm with a lot more pigs. Farms which sell 10 to 15 pound wean pigs to nursery grow finish farms. And then we have wean to finish. This is where farms get wean pigs and grow to market pigs. So this is where they go from wean and up to sell for market. So we have mating systems. We have pen mating, where one of more boars are placed with a group of females. This is an older system for pastured pigs. It requires little labor but less information of when or if the sow was actually bred. So then we have hand mating, so one boar plus one sow. More labor but ensures mating occurred. And then we have artificial insemination, which is the most preferred method. So extended bore semen is used to inseminate a female using a pipette. Moderate labor needed, individual mating occurs, knows when inseminated and how performed. So we have the life cycle of the sow. Sow housing become a topic of controversy over the past few years. Crates versus pens, primarily during gestation. Gestation meaning pregnancy, so they're usually in crates. Some nowadays have a lounging area, but most of them stayed in crates for 3 months, 3 weeks, and 3 days. These facilities have changed from huts and pasture systems to enclosed farrowing houses, generally environmentally controlled housing, or at least supplemental heating for piglets. A piglet weighs about 2 to 5 pounds at birth. So then we have the farrowing crates. Crates are usually placed in a farrowing houses. They are designed to provide a comfortable place for sows to farrow and to protect their both newborn piglets as well as the workers. They minimize newborn pigs being crushed by sows, which occurs when sows accidentally lay on them. They prevent injury to the pigs or workers if the sow's protective instincts cause aggression behavior. So there can be multiple crates in a room. It just depends on the size. And then litters. Litter size average about 10 to 12 pigs per litter. They range. The range is 6 to 16. The number of weaned pigs averaged about 10.72 pigs per litter in 2018. Now is 10.94. Baby pigs are carefully observed to keep mortality to a minimum and ensure rapid early growth and development. The highest death losses of the entire pork production process occur within the first 3 to 4 days after birth. In piglet survival, there are many steps that are taken to ensure survival of all the piglets. So the clean crates and sows prior to farrowing and then maintenance to reduce contamination. We have supplemental heat. Piglets are born with little energy and ability to regulate their own body temperature. Health exams and treatments, so iron supplementation, and then cross-foster to other sows if needed for space. So we have an all-in, all-out practice. So clean, disinfect, and dry rooms between groups of animals. Do not hold back any small pigs. So all the pigs that come out will leave. So sick or carrier animals can transmit disease to other healthy animals when stressed. So then we have the weaned pig. So age at weaning is determined by the facilities and the management. In general, it's less than 21 days, so 3 to 5 weeks average, and are 10 to 15 pounds. sorry, it's more than 21 days, so it's usually 3 to 5 weeks on average, and at 10 to 15 pounds. They move to the next stage of production, so nursery, grower, or wean to finish, where facilities meet the needs of young pigs. Sows have complete lactational estrus. They can be rebred soon after weaning. So nursery or wean pig facilities, most housing for newly weaned pigs has totally slotted floors, which allow the pigs' waste to fall through in a holding pit. The flooring may be plastic coated, wire or concrete slats covered with rubber mats. Slotted floors remain drier and cleaner for a more comfortable environment for growing pigs. Waters and feeders are placed in locations to work with pigs. Normal behavior: eat, sleep, where dry, drink, urinate, defecate where wet. So we have a small nursery pen. So feeding the nursery pigs. Piglets may have been offered feed in the farrowing crate prior to weaning with whey proteins, milk products, grain, and plant and animal proteins. As many as 5 unique diets may be fed to a pig during a 2-month period before it is considered a grower pig. And that's at 40 to 60 pounds. That's when you become a grower pig. Pig which were originally moved to a wean to finish building are simply changed to growing diets at this point. So then we have grower finisher pigs. The phases of production used to be considered separate phases, but now are continuous phases. Less movement and remixing of pigs help maintain good health and production parameters. So then we have the rebreeding of the sow. As stated previously, sows have complete lactational estrus, so they do not have receptivity to the male during lactation. Receptivity to the male equals estrus. Rebreeding the sow depends on the length of the wean to service interval, when she shows signs of estrus, when she is serviced or inseminated. Pregnancy rate can vary, but usually more than 85%, less than 5% fail for farrow. And then the production cycle begins again, so the sows will be rebred after weaning. Gilts will replace sows that have been culled and bred in the same group. This leads to batch farrowing. All the females farrow within 1 week of one another. Management schemes for efficiency and survival of newborn piglets also helps with marketing a uniform batch of pigs. So that was it for a swine lecture. And then we moved on to microanatomy with one of my favorite professors, Dr. Lammers. He's amazing. He's so funny too. He usually doesn't lecture us. He usually teaches us in lab microanatomy, but we had microanatomy of the immune system. So we have functions of the immune system: protection from threats, real or imagined. We have regulation of microbiome, and then the protection of pro and anti-inflammatory mediators. So immune privileged sites that include the placenta and fetus, the eye, the brain, and the testes. We have innate, which means distributed throughout the body, so cannot be specific. They need to call for help, versus adaptive immune response, which have to do with T cells and B cells. So innate are the neutrophils, the macrophages, the complement, the natural killer cells, and the gamma delta T cells. You probably heard about me talk about this in immunology lectures too. So T cells, you have the cytotoxic T cells, which are meant to kill, and then we have the T helper cells, which are influencers. And then we also have the B cells, which produce antibodies. Structures associated with adaptive responses, they're all produced in the bone marrow, then go out to these different locations. So we have the lymph node, produced by lymph. We have the spleen, which has to do with blood. We have tonsils, which is basically the keeper of the gate. We have the mucosa-associated lymphoid tissue. It gets associated lymphoid tissue, so that's GALT. And then we have the bronchus-associated lymphoid tissue, which is known as BALT. Thymus is where T cells are made. And then we have the bursa of Fabricius, where B cells are made. But yeah, all in the bone marrow. So then he showed us the lymph node, which is here. So we have the subcapsule, which goes around it. We have the cortex on the outside, and then the medulla in the middle. So afferent goes in and then comes out. So the lymph node is less dense in the medulla. So he showed us different pictures here. So the follicle, so we have the lymph node. Follicle is kind of like the nightclub, where everything comes in and meets each other. They either like or dislike each other. They send out signals. They have to do with proliferation, so going to go out and secrete or kill stuff. So this is where B cells and some T cells are. So the lymph node, medullary sinuses. So the cells all go back to where they came from after they deal with immunity stuff. So then we have the spleen here, vascular schematic sampling. So sampling, why would we sample blood? Why? To determine bad stuff, because there's bad stuff in blood like bacteria, viruses, and parasites. So we get blood flow into the spleen. We have smooth muscle that is located in the spleen. The spleen contracts in order to send out extra red blood cells. And then barbiturates, why spleen is big and a lot of necropsies. This is due to the barbiturates. The spleen dilates with more red blood cells in the spleen. And then we have the spleen up close here. We have white pulp and red pulp. There are two germinal centers. There is a difference. So the top one here in this picture has been stimulated and it's producing more B cells, whereas the bottom one, if you see this tiny one, has not been stimulated yet. So we have the white pulp, which is also known as the lymphoid portion of the spleen. I don't know if you guys can see it, but it's right there. So then we have the ileum, where we sample intestinal contents. So that's what the ileum looks like. Oh, hold on. Okay. I accidentally touched my screen, and sometimes it doesn't like it because it's touch screen. So then I have to go get my Word document back. So then we get into the tonsils. So these are sebaceous glands, and they have stratified squamous on top. They need a thick protective surface, and then we have the lymphoid tissue, which we collect this for to do with tolerance. So then we have the tonsil up close, less dense in the middle, so terminal center. And then we have the thymus, where it said T cells are made. So if a T cell responds to a self-antigen, it is then eliminated. So this is where we get rid of most T cells, because if they attack the self-antigen, then they are destroyed. And that was it for that lecture. So those were my first 3 lectures. I still have 2 immunology, which I'll have class here in about 30 minutes. But I'm going to take notes online. So I will talk to you guys about those lectures after I actually do them. So, hi guys. So to finish my lectures for today, I did go over them. I'm still missing 1 lecture, but I'm going to try to explain it to you because I do have the PowerPoint and I converted it to a Word document. I just haven't watched the full lecture on it because it hasn't been posted yet. But we're going into Immunology 7, which has to do with acquired immunity. So we're dealing with B cells now and antibodies. So our objectives for acquired immunity are to understand types, classes, isotypes of antibodies and their functions, to learn the physiological and pathological contribution of antibodies, and to understand B cell surface markers, and to learn the T cell derived cytokines that affect the B cells. So what types of cells secrete antibodies? We learned this when we got the intro of immunology, so that would be B cells. They're actually named after a chicken due to a bursa in chicken, so bursa with a B means B cells, and then T cells of course come from the thymus, which also begins with a T. So then we have the physiological importance. Antibodies are important for survival. Antibodies bind to and neutralize toxins. And toxins mean, so antibodies neutralize toxins. This would include like tetanus, microbial toxins, etc. Toxin and a toxin antibody complex cannot bind to target cells. So you give an example of a rusty nail. So if you step on a rusty nail, you have a tetanus vaccine, so your body should already have antibodies created in order to fight against this toxin. So once you step on the nail, you have your antibodies already, so those antibodies should be programmed and then, or signaled, I guess, and then your body will recognize this toxin and then it will get rid of it. So antibodies bind to extracellular bacteria and viral particles, so in the blood, and inactivate and prevent adherence and infection. So you have an antigen-antibody complex, it recognizes it's recognized by macrophages. So the FCR receptors, or also known as FCR, internalized and eventually degraded. This coding of antigen by antibody is called opsonization. If you remember, we talked about that, opsonization being a natural catch-up. So additionally, antigen-antibody complex can activate complement system, which distort antigens by lysis. Antibodies bind to aged and damaged cells. The macrophage have SC receptors, so FCRs, which phagocytize antibody-coated damaged cells and aid in clearance of old cells to keep the cell numbers in body optimum. Homeostatic mechanism that gets rid of dying cells. These cells are also cleared out by apoptosis. Antibodies bind to dying cells. So they are marked as old cells. The FC receptors bind to these antibody old cell receptors, so that becomes a product together, and it sucks it in. Macrophages chops it up and clears it out, basically. So this recycling the old cells in order to create new healthy cells. So it gets all the good stuff out of the old cell, like nutrients and whatnot, so it can form a new cell. The pathological role of antibodies is when B cells become tumorigenic, and cancer results in myelomas. Antibodies against self-antigens, so autoantibodies destroy self-tissues, so this would be autoimmune disease. And then antibodies provoke exaggerated inappropriate response, so the IgE creates this problem. This would be like allergies. Alright, that's why you don't drink that much. Sorry, she always drinks too much and decides to choke. Yeah. Anyway, so what's a myeloma? A myeloma is a plasma cell tumors. They're also called, which are called myelomas. They arise from single precursor cell or clone. So secrete homogeneous immunoglobulin of any class called myeloma protein. You will not actually find plasma cells in the plasma. They are actually found in tissues. So he's like, whoever named these, yeah, it didn't make sense. If you find these in circulations, that's not a good sign, so it means there's a tumor somewhere. So then we get into serum electrophoresis. Antibodies are primarily found in the gamma region. So here's this. So this is normal antibodies right here, and this is if you have a disease, so it's way up. So that's when you can tell you have a myeloma protein by this test. So we have myeloma cases. So the active osteoblast, which causes the density of the bone to decrease. Osteoclasts are eating up the bone. So they make lots of antibodies, but they're worthless antibodies in these myeloma patients. They are immune deficient. They only make one class of antibodies against, again, useless antigens. In jaundice cases, that means the yellowing, I'm sure you've heard that. Autoantibodies against red blood cells bind to the red blood cell and destroy them through complement-mediated damage or opsonization. They facilitate phagocytes to engulf better antibodies, so the good antibodies. So this is where, so autoimmune disease means where your body's basically attacking itself. So the antigens are attacking, like T cells attacking self antigens. That's bad. So yes. So then we have systemic lupus, erythematosus, which is also known as SLE. Human lupus, you would get a butterfly rash on your face. In canine lupus, you would get major arthritis, skin lesions in different parts of their body for canines. They target the skin and kidneys. And then you have rheumatoid arthritis, which you'd have deformed digits and joints. Autoantibodies against synovial membrane and joint tissue. This is the rheumatoid factor, which induced the arthritis. It's very painful, and it tends to be bilateral as well. So then we got into antibodies and allergies. So hypersensitivity type 1. So type 1 hypersensitivity is an allergic response. It usually has to do with IgE and eosinophils. Note for generation of IgE antibody, Th2 cell cytokine IL-4 is essential. So if you're stung by a bee, this antibody will bind to mast cells. Mast cells have a ton of granules. And as soon as an antibody binds, they degranulate these molecules that come out of the mast cells and act on the blood vessels, and increases the permeability a lot. Hold on, I have to retype something. But anyway, so you get this allergic response. And then you have IgE-mediated allergic response, which you saw in the horse there. And then we have B cell development and differentiation. So the primary lymph node organ is the thymus. Like I said, that's where T cells are made. But secondary are the lymph nodes and the spleen. These are all mature B cells once they reach the lymph nodes and the spleen. So here, again, we have primary lymph nodes. So there's where development occurs. So no antigen interaction. But then we have secondary lymph node organs. So no further development of lymphocytes. They're the site of antigen interaction. So that's where the spleen and lymph come in. Here, we're still in the bone marrow. So we have the stem cell, which yields to a pre-B cell. So cytoplasmic U chains, and then it becomes an immature B cell. It is membrane bound by IgM and IgD, not O. And then it yields a mature B cell. So the antigen connects here, as well as the class switch. The mature cell then becomes either a memory cell or a plasma cell. Yeah. So then note that bone marrow is both a primary and secondary lymphoid organ. So B lymphocytes, B cells have dual functions. Like I've said before, they can secrete antibodies production and antigen processing and presentation. Antibodies play a critical role in defending against extracellular pathogens. Antibodies cannot penetrate inside a cell, therefore not effective against intercellular pathogens. So antibodies exist in two forms. So secretory, so it's detected in fluids such as the serum, which is the main one, the peritoneal cavity, and mucosal fluids. But also they exist as membrane-bound surface. They're important for specific binding to antigens. So we have antibody features. B cell is a round cell, has a lot of receptors, which increases its chances to bind to an antigen. It exists in two forms. So again, membrane-bound on B surface as B cell receptors, so BCR. There's about 200,000 to 500,000 identical antigen receptors. T cells only have about 30,000 TCRs. So each BCR, so B cell receptor, is specific to only one antigenic epitope. Two, the secreted form antibodies or immunogens are found in bodily fluids. Secretory free form of antibody is found in the blood, so serum and plasma, and body fluids as immunoglobulin. They neutralize antigen and assist in their removal. Antibodies recognize free antigens, so B cells recognize free antigens, whereas T cells cannot. They do not require combination with the MHC, unlike T cells. Antibodies are specific. They bind specifically to the antigen, which induced antibody production. They cannot penetrate the cell. And then we have antibody structure. So they are glycoproteins made of 4 polypeptide chains. They have a flexible Y-shaped structure. So they look like this. So they have 2 identical smaller chains, also known as light chains, and then they have 2 identical larger chains. Those are known as heavy chains. There's a hinge region here that allows it to move. Antigen can only bind to the top portion, variable because they have variable regions and portion changes. So up top, they have a fragment antigen binding, or also known as FAB2. At the NH2 end, there are 2 antigen binding sites per antibody, and then they also have a fragment crystallizable, or FC region, where it interacts with the effector molecules, so the complement cells and macrophages. So that's at the other end, the bottom end. Antibodies recognize shape of antigens. Shapes of antigens, antigens are known as AG, and CDR, remember, is complementary determining region of antibody. Each V region is folded in such a way that 3 CDRs come into close contact with the antigen, this creates unique seats for the antigen to sit on. So again, that promoting specific binding to antigen that is induced by the antibody response. So then we got into the next lecture, which he started. So we just continue on. We have antibodies or antibody classes or isotypes. So they're based on heavy chain characteristics. Antibodies are divided into 5 classes. You have IgM, you have IgG, you have IgA, IgE, IgD. So we're going to go over IgM first, which is 5 stitched together by J chains. So it has 10 opportunities to bind. So the first antigen or first antibody, so IgM is the first antibody to appear in circulation. It's predominant in primary antibody response. It's the largest in size. Membrane-bound form is a monomer. Secretory form is a pentamer. So like I said, they have 5 attached together. With the J chain, seen primarily in the bloodstream and the second highest in serum immunoglobulin levels. So its functions are to agglutinate antigen and to neutralize the antigen. So classes or isotypes are most abundant. So we're getting into IgG. So they're the most abundant, they're a monomer, they're the smallest antibody, can cross placenta in some species, and can move out of the bloodstream and is found in tissue, blood vessels, and other secretions. Their functions are to agglutinate antigen, to neutralize antigens, and it's important in inflammation. So then we have the IgA, which is a predominant antibody in mucosal tissue. It's actually the only antibody that can survive in mucosal tissues, while the others cannot. It has a secretory component that protects it from digestion by proteases. Enzymes cannot attack IgA where they attack the others, so they can survive in very hostile mucosal tissues. So again, it has a secretory component which allows IgA to appearance in secretions and protect it from digested by proteases. It prevents adherence of pathogens and antigens in general to mucosal surface and agglutinates. It tends to be synthesized under intestinal, respiratory, reproductive, and other mucosal surfaces. So then we got into IgE. It can't bind to mast cells. It has to have a Fc, so it has an extra domain. But we don't want this for allergies. We don't want IgE. We want IgG instead because they can bind to the mast cells directly. But IgE is a monomer. It's heavier than IgG. It's bound to mast cells and basophils, so that's tissue and blood respectively, through a receptor Fc, like I just said. So it says it's important in allergic reaction, but we really just want IgG. IgD, he didn't really go over it, but it's a monomer similar to IgG, is BCR bound only. Secretion is negligible. It's found in humans, rats, and mice, but he said this wasn't important, so we don't really need to know it for the test. So then we have a primary immune response. So we have the short lag period, and then we have the IgM secreting cells that proliferate, and then we have the death of the the LGM secreted cells. So only a few cells survives, and these are the memory cells. So I think we went over that in the first lecture, actually, where antibodies are stored as memory cells. So then when, so that's the first response, but when there's a secondary response, they know they have antibodies in the memory cells, so the memory cells are stored. So when it comes in, the body is able to recognize the second time go around. So again, we have the primary immune response, but then the secondary immune response recognizes it already. So it has a short lag phase, and the body is ready to attack at this point. So we got into class switch. So why have class switch? Reasons for class switch. So IgM to IgG. IgG is the only AB which can penetrate placenta and blood vessels. IgM to IgA. IgA is the only antibody that's survival in mucosal sites that is enzyme rich and acidic. IgM to IgE. IgE induces allergic type 1 hypersensitivity reactions. So we have antibody isotopes or classes, which there's a whole table on this. I'm not sure if we need to know, but it just goes over basically the summary of everything that I just went over. So class switch antibodies retain specificity. So during an antibody response, class switch changes in allergy class occurs in standard sequence. As immune response progresses, B cell switches in presence of Th1 or Th2 cytokines and co-stimulatory molecules to one of the major secretory classes, so either IgG or IgA or IgE. Fab does not change during class switch. So class switch means, I'm guessing, it changes in allergy class, so you can change from different allergy classes. IgM, anti-canine distemper antibody, after switch to IgG, will retain specificity for canine distemper antigen. So antibodies exist in two forms, like I said. We got into secretory and memory-bound surface, so B cell receptors, also known as BCR, are antigen receptors. So here, as you can see, IgM secreting B cell can do a class switch to become IgE, IgA, IgG, or IgD. That's when Th1 or Th2 cytokines yield to the IgM secreting B cell. So then we have B cell receptor, BCR recombination. BCR determines the specificity of B cells. Example, BCR binding to salmonella antigen is different than BCR binding to E. coli. And I think I talked about that before in one of my sitcoms, so I'm not going to go through that again. But then we have membrane-bound B cell receptors. So BCR is the antigen receptor on B cells. It is membrane-bound. Each B cell may have identical 200,000 to 500,000 BCRs, like I said before. All BCRs on an individual B cell have the same specificity, so antigen binding site. All B cells express, so B cell receptors, they bind specifically to an antigen. CD19 binds to C3D, decrease the threshold for B cell receptor signaling pathways. CD40 is a receptor for CD40L or CD154 on activated T cells. So the interaction of antigen processing B cell with Th2 cell, we have that here, which I've talked about before with Th2 cells and the B cell. So Th2 cytokines act on B cells. Th2 cells secrete cytokines that initiate B cell activation and differentiation. IL-4 induces class switch, so IgM goes to IgE. It enhances MHC class 2 and FC receptor expression. IL-5 acts on activated B cells to differentiate into plasma cell. So antibodies secreting cells not present in the plasma. And then we have IL-13, which has the similar functions as IL-4. And then IL-6 is the final differentiation of activated B cells into plasma cells. So basically a Th2 cell yields to an IL-4, 5, 6, or 13, which then yields to an IgM B cell, which then yields to a class switch from an IgM to an IgE, or whatever it's becoming. So we have the summary B cell surface markers. So we have B cell receptors, we have the CD19, the complex that binds with complement C3D. We have CD40, which binds with the CD40L on T cells. We have CD86, which binds with the CD28 on T cells. We have the MHC class 1 and MHC class 2, antigen processing and presentation. And then we have the CH2 cytokine receptors such as IL-4R. So how does the immune system maintain flexibility to react to diverse antigens? Well, here we go. Mutation. MHC has, yes, the highest rate for mutation. So T cell receptor, no, and B cell receptor, yes. And then for recombination, MHC, there's no, T cell receptor, yes, and then B cell receptor, yes. So that's just basic summaries of what I just talked about. But that was the last of the immunology lectures before our exams. So wish me luck. I have OSCEs tomorrow, which is our clinical skills exam. It's only like 36 minutes long, but we literally have people staring at us and we have to do something and talk our way through, just like a regular veterinarian. And then if you don't pass, they can kick you out of vet school. So you get 3 chances if you don't get things right. So there's 5 stations and you can fail a station, and then you have to retake it probably on Saturday of whatever stations you did not pass. But if you don't pass it the second time around, you go into a third time, and then if you don't pass it then, you have to go before the board and tell them why you deserve to stay in vet school. So it's pretty scary. But anyways, so wish me luck tomorrow. I have my OSCE, so get that out of the way, and then I'll be studying for my lecture exam on Thursday. But I hope you guys have a good night.